Yes, my name is Nora Romero. I'm a PhD student at the Packard Lab in New York University, and I'm very happy to be talking to you about the bee genus Liphanthus. My presentation will follow standard format of preliminary research, give you some findings and future work at the end. My PhD research deals with the revision, phylogeny, biogeography, and natural history of Liphanthus. In this talk, I will be talking to you about progress I made with the phylogeny part of the study. But before we get into details, I'd like you to get to know my bees a little bit. So all this here is Liphanthus, is a relatively poorly known group of bees. They're quite small, but show remarkable sexual dimorphism. The one here on the left is a male. It's a male of Liphanthus abulosus. It's one of the largest species, and it's only 6 millimeters in length. Females can be a little bit larger, but less colorful than the males. And, you know, as we can see, it looks like a wasp, and it actually, the name Liphanthus is an anagram for Phyllanthus, which we saw in Thomas Onufrika's presentation just a little bit earlier. In terms of higher classification level, this is where Liphanthus sits. And in terms of biogeography, or in terms of geographical distribution, I should say, this discovered life here shows that Liphanthus has mostly a Chilean distribution, but certainly we expect to find many more species in the Andean region, and I will talk about that later on in my presentation. This beautiful calibrated tree here is a fragment of the most recent phylogeny of the subfamily Panurginae. So we can see here that in the Protandrenidae, Liphanthus started diversifying around 10 million years ago, and its closest relatives are Paracyanithia, Parasaurus, Astropanurgus, and the Pyraphyletic Raphidilis. In terms of apomorphics that separate this genus from other groups, is the most distinctive one is on the stigma of the forewing. This stigma, as we can see here, is very narrow, almost as narrow as the prestigma. And the inner side that falls in the marginal cell is concave or flat, whereas other groups, this side is strongly concave, and the stigma itself is wider than the prestigma, clearly much wider than the prestigma. Just talking a little bit about the taxonomic history. The first revision of the genus was done by Luisa Bruce and Haroldo Toro in 1983. They described 25 species and erected 7 subgenera. Since then, more species have been described, including some work by Mir Sharifi and the Packard Lab. Most importantly for this presentation is that from the 49 described species, only 30 have been placed in a subgeneric category, and 19 still need placement. So aside from described species needed placement, there's also the situation of Liphanthus being more speciose than previously thought. So Packard and Bruce found in 2016 that they discovered 47 species in the genus just by means of DNA barcoding. We have been finding more species just with morphology. My estimate is that there are around 32 undescribed species that has no placement, and as this nice looking neighbor joining tree of Liphanthus sequences shows, that the colorful sequences are correspond to the described species versus the black ones corresponding to the undescribed species. Home take message if you didn't get it is that most species in the genus net subgeneric placement. So the objective for my presentation was to present you to my results of the phylogeny, which have been giving me insights about then some subgenera that needs to be redefined or erected, the placement to subgenus for species that have not a placement, and a very one specific objective that I have was to test if the genus Liphanthus sensu stricto and the subgenus, Pseudoliphanthus are reciprocally monophyletic. Talking a little bit about my methods, this phylogeny was done at the subgenus level. All subgenera were included. I took 2 male exemplars from each subgenus, except for Pseudoliphanthus. We have 4 species in that taxon, and 1 species for Neoliphanthus because it's a monotypic subgenus. For my outgroup, I chose Parasaurus, and the next steps I will be giving you, I will be describing to you more in detail. Now, this is the fun part of the research. I get to describe to you some of the characters, the morphological characters that I explored in the males. The image here on the left, it shows you an example of both alternative states. In the following pictures, I'm not really going to say it. So here we can see one of the characters that I use, the interocellular distance in relation to the distance to the eye. We can see here both alternative states being greater here, smaller here. Now for our star species of Liphanthus sabullosus, again, the male has the outer subantennal suture strongly arcuate. The alternative state is being just straight. The vertex of the species is convex. Alternative states is being strongly concave or flat. Facial fovea, it's almost rounded as opposed to very elongate. The clypeus here, we can't really see it, but it's strongly convex. In other species, the clypeus, it's flat or even concave. The labrum also has some variations. This one here of Liphanthus genamro, it's the labrum is slightly convex. It could be also flat and can have or cannot have a transversal carina. So the mandible of this species also has a tuft of hairs. It can be or cannot be present in other species. More fun characters. This is the forewing. It can have 3 submarginal cells, or just 2. Mesoscutum has some variation as well. This one here shows relatively on a sculpture mesoscutum and very shiny. The hand tibial spurs of Liphanthus genamrum and other species are strongly curved, as opposed to just slightly curved or even not curved at all. The metasoma of the males is it's remarkable that can have coloration, yellow coloration or not. And my favorite character for the males is that the sternum 2 can have a great modification in the form of a spine or some sort of protuberance. So this is suspected to have a stimulating role in mating, of course. This is what we suspect, because what else can it be, right? So I compiled 60 male characters for 26 Liphanthus species and my Parasaurus as an odd group. I compiled this in a matrix that I analyzed with maximum parsimony through TNT and with CLADA programs. So this is our own beautiful phylogeny. My raw data generated one most persimmons tree. This is the tree with the subgeneric classification from Luisa Ruz and Haroldo Toro. This is the colors here represent the subgenera, with the pink color representing the unplaced species. We recovered two monophyletic groups, being Liphanthus and Liphanthus, and some non-monophyletic groups, including, so for the rest of the subgenera. Now this is the same tree, but with my interpretation and the incorporation of the new species. We recovered this monophyletic group that will be erected into a new genus with 3 described species and 1 new species. Now, the monotypic Neoliphanthus will be merged into Leptophanthus as it represents 1 monophyletic group. Melaliphanthus has gained 1 species. Phasganuventris was unplaced before. And Xenoliphanthus has gained 1 species here, 1 new species. Now for my specific objective of proving if they were reciprocally monophyletic Sensus strictus and Xenoliphanthus, because Sabulosus and Barbatus representing Sensus stricto is nested within Xenoliphanthus, we conclude that they're not reciprocally monophyletic, so it's going to be just 1 subgenus. Liphanthus Sensus stricto, because it was the first one described. So my conclusions is that my phylogeny divided the genus in 2 major clades. Here we erected one new subgenus. We tested the monophyly of some subgenera. We placed 4 new species in subgeneric category. Some things that we need to do is to resolve the placements for the new species, including all the new species that we haven't included in this phylogeny, and also this new species here. And I'm confident that with adding more characters, adding more data, we'll be able to resolve the paraphyly of Tricoliphanthus here that shows in red. Yeah, for future work, I will get to play with more fun characters, including genitalia, of course, and some new structures from the tentorium and mouth parts. That's going to be done for both males and females. And in the horizon, there's also, would like to make our own beautiful calibrated tree with Liphanthus species using ultra-conserved elements. We'll be having an almost complete taxon sampling for that, which is very exciting. For future work, I'm going to be exploring more of the Andean region, trying to get more insights on the biogeography and also the natural history. We'll analyze some pollen, some parasitism aspects of Liphanthus. So a lot of field works, museum for those species, for those countries that don't allow a lot of collecting. And yeah, it's going to be a lot of fun. I would like to acknowledge my lab and other people who have helped me, to my amazing supervisor. And I kind of love the idea of thanking the bees. So there it is. The cat couldn't show up, but that also would have been great. Thank you very much for you. And with that, I'm through. Please, if you have any questions, please let me know.